Yesterday we discussed the cloning strategies, and as a part of that, the genomic DNA library construction is already discussed in the last session. Now, our next topic is cDNA library preparation. cDNA libraries, they are actually, it is a library of a population of bacterial transformers or phage lysates, in which each mRNA isolated from an organism or tissue is represented as its cDNA insertion in a plasmid or phage vector. So that is the standard definition that can be given to a cDNA library. That means it's a population or a collection, a library of a population. That's a population or a collection of bacterial transformants or phage lysates, which means the vectors, in which each mRNA isolated from an organism, or all the mRNAs isolated from an organism or tissue, is represented as its cDNA or complementary DNA insertion in a plasmid or phage vector. So we are constructing a collection of recombinant DNA constructs with the plasmid or phage vectors, which is incorporated with the complementary DNA prepared from all the mRNAs isolated from an organism. So that is the definition for a complementary DNA library or cDNA library. Complementary DNA or cDNA libraries are generated by a reverse transcription process of the mRNA. cDNA is a representative of mRNA population, and it is therefore reflects the mRNA levels and the diversity of the splice isoforms of a particular species, which means cDNA is a representative, or it is actually coded from the mRNA population. For every cell type, time to time, there may be some particular mRNAs that is expressed at a particular time in response to its growth stimulus and other environmental factors. So, a cell type, for example, a blood cell, or say insulin preparing cells of the islets of Langerhans, or some other particular type of specific type of cell, which actually expresses a particular population of the mRNA. You know that there are cell-to-cell difference in the gene expression patterns. So, which means a specific cell type which is producing a particular type of the enzyme or a particular, say, a derived cell, like the pancreatic cell or some other cell type, which is specialized for performing a particular task, will have its own population of the mRNA that is produced within the cell. So, based on the environmental conditions, based on the genetic makeup, based on the different hormonal and other response elements, and also based on the cell type in which it is expressed, there is difference in the mRNA population of each and every individual and individual cell type. That's why there are different splice isoforms in specific tissues. You can't expect the same mRNA population in a cell type like a fibroblast cell and the pancreatic cell, which will have different functions. So the population of the mRNAs which can be isolated from that particular cell type will be different based on the nature of the cell type or the tissue type and other environmental stimulus or other growth promoting factors that is influencing the gene expression. So the cDNA actually, it is a representative of that mRNA population, and that is specific for that particular cell type under a particular set of conditions. So even the conditions, the expression conditions is also important. For example, the same bacterial population of E. coli will have a particular set of mRNAs which is produced in the presence of lactose or in the absence of lactose. Because we know the expression of the lac of protein in which there is a differential expression. So based on the environmental stimulus or there's some nutrient availability or some other factors, the mRNA population that is coded from that cell type will be varying. So that's why we are saying that the cDNA, it's a representative of the mRNA population and which reflects the mRNA levels and the diversity of the splice lysosomes and our every particular cell type. The first stage of the cDNA library reconstruction is a synthesis of the double-stranded DNA using mRNA as the template. Now, in the preparation of the cDNA library, the reverse transcriptase is the enzyme that is where we have a DNA-dependent DNA polymerase, which is a normal DNA polymerase, which depends on a DNA template and produces a DNA. So that's a DNA-dependent DNA template, a DNA polymerase. Whereas reverse transcriptase is another enzyme which depends on an RNA template and produces a DNA copy. So that's a DNA-dependent RNA polymerase. It is discovered by Chemin and Baltimore in 1970, and it's obtained from avian microblastosis virus, that is AMV. It requires a primer. So just like other DNA polymerases also, it requires a primer, whereas the RNA polymerases may not require that one. So here we have the reverse transcriptase, which is a DNA polymerase, and that RNA-dependent DNA polymerase requires a primer with a 3' OH group. And it uses the oligonucleotides, that is poly T, in eukaryotes as a special trick for it can use the poly T, which is present. Poly T as a primer. The poly T is a primer that can be given to all eukaryotes because we have poly A tail at the end of all eukaryotic mRNAs. For example, if you require a primer with a free 3' OH, a poly T primer that is constructed there is a poly A tail at the end of all eukaryotic mRNAs, which will bind with the poly T primer which is provided, and it will amplify. So a poly T commonly used or generally used will amplify all eukaryotic mRNAs. Whereas we require special tricks for prokaryotes and other viruses where such a poly A tail is missing with the mRNAs. Now there is a problem with cDNA preparation. There are some problems reported. One is the incomplete copying of the mRNA by reverse transcriptase. And that means if the mRNA is incompletely copied by the reverse transcriptase, 5' end of the mRNA will be missing. So that means starting from the poly A tail, from that end, it will start copying. So during the copying to one end, if it prematurely terminates, for example, the mRNA may have a length of 1,000 base pairs or 750 base pairs, and during the process of initial stage of the reverse transcription, the transcription stops abruptly at about 500 base pairs. So it produces an incomplete product of the reverse transcription product. Actually, we are expecting a product of, say, 750 base pairs, but the premature termination of the reverse transcription process creates shorter mRNA, shorter cDNAs, which will have a missing 5' end of the mRNA. So that end is not completely copied. Then incomplete copying of the cDNA single strand, that is a 3' end of the mRNA. So first of all, the copying of the initial strand by reverse transcription process is done. Then the second strand is copied back during the process of cDNA synthesis. So first of all, using the RNA template, the RNA dependent DNA polymerase of the reverse transcriptase copies the first strand of the complementary DNA. And premature termination of that one results in missing 5' end of the mRNA. Again, if it is completely copied, then the second strand of the DNA is copied back, and that strand is synthesized. If that second strand synthesis is incomplete, then it produces cDNA copies with the missing 5' end. So anyway, incomplete copying or premature copying of both the strands of the cDNA will result in shorter strands or incomplete cDNA copies, which is useless in your case. So that is one of the problem. And the nuclease is used for cleaving the hairpin loops, and we will see how it is done. And there is a hairpin loop during the copying, which is created at one end, and you may require S1 nuclease for cleaving that one. So it may nibble away or cut a few bases from one of the end. So if you are trying that type of nuclease treatment, some of the bases at one end may be missing. So this is some reported problems with the preparation of the cDNA fiber. Now, when we come to the isolation of the mRNA, the first step is the isolation of the total mRNA of the cell type. So every cell at a particular time, at a particular growth condition, will produce a particular set of mRNAs. If the environmental condition changes or the growth condition changes, then some of the mRNAs may shut down and some other population may start producing. So the isolation of total mRNA, it is actually dependent on the different growth conditions and other things. So at a particular or a specific growth condition and a specific environmental condition, and that population of the cells will produce a group of mRNAs in response to that one. So you can isolate all the total mRNA population from that cell type. Then chromatography using polyucephalose or oligo-T-cellulose is one of the purification methods that can be used for isolation of the total mRNA. So a chromatography column, which is filled with the cephalose beads linked with the poly-U residues or oligo-T residues or oligo-T-cellulose residues. So cellulose linked with oligo-T or cephalose beads linked with poly-U residues will be loaded in a column, a chromatography column, and that column can be used for separating this total mRNA. Because, as you know, all eukaryotic mRNAs will have a poly-A tail at the end. So if the chromatography column is packed with poly-U or oligo-T cellulose bound beads, then the mRNAs will stick on to that poly-U or oligo-T because there is an AU or AT specific pairing, and which will hold the eukaryotic mRNAs and all other mRNAs, all other materials that is not bound with the poly-A will be eluted. And that can be by changing the buffer. These bound mRNAs, eukaryotic mRNAs, can be eluted from the chromatography column. So a chromatography column with poly or oligo-T can be specifically used for isolation of eukaryotic mRNAs because it will hybridize with the poly-A tail at the end of all eukaryotic mRNAs, and it will be retailed within the chromatography column. And after washing, you can elute these poly-A tail containing eukaryotic mRNAs in the second step. So another possibility of eukaryotic mRNA isolation is a density gradient centrifugation, which will separate the mRNAs based on their density or the size, and that is one of the methods that is used. Then another method is use of antibodies to the proteins of the mRNAs and precipitation of the polysomes. And you know that the mRNAs usually it will start translating with binding to the ribosome assembly, and multiple ribosome units will bind to the same mRNA and start translating. So that is a polysome complex in which a single mRNA is bound with multiple translated ribosomes, and peptides of different length will be attached to each and every ribosome. So the first ribosome, which is actually moving ahead, will have a longer polypeptide chain attached to it, whereas the last bound ribosome will have a shorter polypeptide because it's only beginning to start the translation. So anyway, during the translation, you'll have an mRNA which will be bound with the multiple ribosomes, and each ribosome starts translation, and the ribosome initially bound and leading ahead that will have a larger polypeptide which is produced, and all these complex mRNA, ribosome, and the translating polypeptide. All these things will have it will form a complex, and that polysome complex is there. So if you are targeting an antibody which is specific against the polypeptide that is produced or that is being translated, and that results in precipitation of this entire complex of the polysome. So if you can develop an antibody and treat such polysome, translating polysome, then the antibody will bound to that nascently created or the nascent polypeptide that is being translated, and the available region will be bound with the antibody, and which will settle and precipitate that polysome complex. And from that one, the mRNA can be, or the specific mRNA. For example, if you are targeting a particular mRNA, if you want to isolate a particular mRNA of a particular protein, then you can use such antibody binding methods against the polysomes, which will precipitate the specific mRNA against that protein, and that can be isolated, and then you can go for a cDNA synthesis from that mRNA. The another one is isolation of the mRNA from a specific tissue where it is expressed, which means you are looking for cloning a cDNA from pancreatic cells, and you want to produce insulin out of that using a cDNA clone. So you know that insulin is produced by pancreatic cells, so you know where to look. It is a specific cell type where it is expressed, specifically expressed. So you need to isolate the specific tissue type where it is expressed, and processing that one will produce a large number of population of the mRNAs of the concerned protein. Whereas if you are looking on another level, for example, brain cells, you will not get it because it is not expressed there. That mRNA will not be there. So the mRNA of a specific cell type can be isolated from the specific tissue where it is expressed. Another one, use of this technique for the expression of a eukaryotic gene in prokaryotes. So you can use these techniques for expressing a eukaryotic gene in prokaryotes, because normally eukaryotic genes cannot be expressed in prokaryotes because eukaryotic genes will be structurally different. The DNA will have introns and exons. It needs to be processed. It requires splicing and the joining of the introns, so removal of the exons, removal of the introns and joining of the exons to produce a functional mRNA. And also it needs to be placed under the control of a regulatory element which is useful for eukaryotic genes, but which is not available in normal prokaryotes. So a simple cutting and pasting of the gene, eukaryotic gene, as such, the chromosomal segment into the prokaryotes will not express the concerned protein in prokaryotes, which requires cDNA preparation. cDNA preparation means the mRNAs of eukaryotes were actually prepared by all the post-transcriptional modifications, including splicing. So, which is actually ready to be translated. If you are producing a copy of that cDNA from that eukaryotic mRNA, then eukaryotic regulatory mechanisms becomes irrelevant. If you place it under the control of a prokaryotic promoter, then it will express because it's a simple DNA fragment. Eukaryotic regulatory elements and eukaryotic splicing mechanisms will not matter because the cDNA is prepared from an mRNA which is ready to use, and placing it under the control of a eukaryotic promoter will simply amplify that one, and it will be expressed. Whereas a eukaryotic gene, without such processing, post-transcription modifications, if you are placing it in a prokaryotic system, it will fail because the prokaryotes doesn't know how to translate it and how to transcribe it, how to modify or modify it after the post-transcriptional modifications and the splicing, everything. That mechanisms were not available in prokaryotes. So for cloning a eukaryotic gene in a prokaryotic system, always you require a cDNA library. So from the cDNA library, you have a functional mRNA from which you copy the cDNA, then place it under the control of a prokaryotic promoter itself so that it will be expressed in that system. So this is some of the features of the cDNA preparation, and we discussed the isolation of the mRNA and some of the specific techniques that is used for the isolation of the mRNA. Now, we have an example here. We have a series for interferon, blood clotting factors, HC, and several other mRNAs, eukaryotic mRNAs, were expressed in bacteria and including insulin in bacterial systems using such techniques, cDNA cloning techniques. Is it clear?
Yes, sir.
Hello? Okay, fine. Can I continue? Any questions?
Yes, sir.
Okay. So now we come to the different strategies that can be used for preparation of a cDNA library. So here it is shown as an early cDNA cloning strategy, which is involving a hairpin primed second strand for the cDNA synthesis and a homopolymer tailing to insert the cDNA into a vector. Here we have a PBR322 vector, which is shown here in circularized form, and also we have an mRNA from which the cDNA needs to be produced. Here the vector can be cut open and linearized by PSD1 restriction enzyme, so it is linearized, and then a terminal transferase for a DGTP or deoxy GTP is used against that linearized vector, which will add poly G at both ends of the sticky edge. So the terminal nucleotide transferase and the concerned nucleotide, specific nucleotide, will add the poly G at the end of the linearized vector. Now the mRNA, from that mRNA, which is having a poly A tail, you are using a poly G primer. Already we mentioned it in the previous slide. All eukaryotic mRNAs is having a poly A at one of the ends. So a specific primer of poly G can be targeted against that one. And you know that reverse transcriptase enzyme, since it is a DNA polymerase, it requires a primer with a 3' OH. And poly T primer can be used against all eukaryotic mRNAs. So it will amplify the first strand of the cDNA, and that reverse transcriptase and alkali treatment. So the alkali treatment will lyse the RNA. So after the synthesis of the first strand by poly T primer and the reverse transcriptase, you will treat it with the alkali. So the alkali treatment will lyse the RNA, leaving the first strand of the cDNA as a single stranded copy. So at one end, the free 3' end of the cDNA will act as a primer. So here you can see that there is a 3' end of the newly synthesized cDNA. So that will act the OH of the 3' end OH of the initially synthesized strand will act as a primer for copying of the second strand of the cDNA. So the DNA polymerase 3 will create, it will synthesize the second strand so that there is a loop at one end. So this loop is created because the initial strand, the end of the 3' end of the initial strand or the first strand of the cDNA, which is synthesized by the reverse transcriptase, will act as the primer for the second strand synthesis by the DNA polymerase. So that second strand is now synthesized, forming a loop at one end. So this single-stranded loop at one end is cleaved by S1 nuclease. And we already discussed the S1 nuclease in the list of the tools used for genetic engineering. S1 nuclease can cleave single-stranded region only. So there is a small stretch of a few base single-strand region that is formed at one end, and that will be cleaved by the S1 nuclease. And in one of the previous slides, we discussed about removal of a few bases as a shortcoming of cDNA preparation. We discussed that such nibbling or cutting of such a loop by S1 nuclease will remove or trim some of the bases. So if you are looking for a very specific amplification of 100% sequences of the mRNA, then this missing nucleotides at one end of S1 nuclease removed by the S1 nuclease may be a limitation. Okay. Now, uh, S1 nuclease will create single-stranded, double-stranded DNA. Then there is a terminal nucleotide transferase which will add poly C at both ends. Now we have a vector which is prepared with poly G at one end, and the cDNA which is prepared with poly C on both ends. And this can be annealed together so that the circular vector is again circularized and formed with the recombinant DNA, which is specifically added. So this can be, again, DNA repair mechanisms, host repair mechanisms will repair and fill the gap and seal it together. So now we have the successful transformant with the concerned DNA, mRNA or cDNA copy, which is inserted in the PBR322 vector. So this is one of the preparation method for the early method or initial basic method of a cDNA library preparation. Is it clear?
Yes, sir. Okay.
Okay. This is another illustration. It works in the same manner, but it's a little bit advanced level. Here we have the mRNA with a poly-A, eukaryotic mRNA. So one possibility is that you are using a primer, just explained as the previous case. For example, you are using a poly-T as a primer, and initial strand is synthesized, then the second strand is synthesized, and for which you are using your DNA polymerase 1 and RNA-ASH, DNTPs. So the second strand of this is synthesized here. So anyway, you want to clone this one. So a simple amplification of such mRNA into a cDNA will not facilitate a successful insertion into a vector. For inserting it into a vector, specific insertion into a vector, you require some sticky ends at both ends, which is not normally available if you are simply using a reverse transcriptase enzyme and producing a cDNA copy. There will not be any sticky ends at the ends. For successful incorporation into a vector, you require sticky ends on both ends. How you can add that? So one possibility is that you can, after the cDNA synthesis, you can add an echo R1 adapter by a T4 DNA ligase, which is a blended ligation enzyme, and echo R1 adapter can be added on both ends and cleaving it with echo R1, and then again the vector can be digested with echo R1, and this echo R1 adapter linked DNA construct can be inserted into the concerned vector, which is opened with echo R1, and now the cDNA library can be constructed. So that's one mechanism where the cDNA is prepared just as in the previous case. You can, in the previous example, we are simply adding a poly G and poly C on, you are attaching poly G on what vector and poly C at the cDNA. Then using that one, you are attaching it to the vector. Here, instead of the poly C and poly G combination, or poly-A tailing or polynucleotide tailing addition, you are adding echo R1 adapters with the T4 DNA ligase on the ends of the cDNA library, the cDNA copy, and then the vector is cut open or digested with the echo R1, and this can be ligated. So the cDNA library, which is inserted with the DNA fragment, can be created. So that's one possibility with a specific restriction enzyme. Now there is another possibility of a linker addition. So in this case, we have this eukaryotic mRNA with poly-A, and you are using a poly-T. But there's a slight modification. Instead of the poly-T, you are adding, you are constructing a primer with poly-T, that's a pairing region, and an additional extension for XHO1, that is a restriction enzyme site. So these sequences, GA, GC, TC sequence, which is shown here, is additionally attached to the primer region. So there is a pairing region for the primer, which will specifically pair with the poly-A tail, and there is an extension of one specific restriction enzyme like XHO1. So using this specific primer, constructors for primer, you are amplifying the cDNA. So initial strand is synthesized. Again, you can add, you can copy the second strand is also synthesized. So second strand, after the synthesis of the first and second strands, you will have an XHO1 site at one end. Just after the poly A and poly T sequence, we have a restriction enzyme site of XHO1 here. So you can use another, okay, methylation enzymes needs to be used because the older strand needs to be protected. So instead of using any other restriction enzyme, you have to protect your strand with the methylation enzyme, DNA methylases. Then echo R1 adapters can be used. So echo R1 adapter is again attached on both ends. Then you use XHO1 digestion and echo R1 digestion. Both enzymes can be used. So here you can see that XHO1 will lies from this point and echo R1 on the other end. So the result will be, if you are treating it or digesting it with both the enzymes, echo R1 and XHO1, one end will have XHO1 and the other end will have echo R1. So this is a specific type of construct that can be used to ligate into a target with the different restriction sites, different sticky ends. So in the previous case, that's a simple joining of echo R1 sites. The vector opened by echo R1 and the construct which is attached with echo R1. So it will specifically add it. But if you are targeting the sequence against, or you are trying to insert this cDNA into a target with two different restriction ends. One end will have echo R1 and the other one will have an XHO1. So that can be used just like an adapter. So this is another form of cDNA library preparation in which you attach two different restriction enzymes on both ends of the DNA cDNA copy. So this is another strategy. Is it clear?
Yes, sir.
Okay. Now this is an improved method for cDNA cloning. And the first strand is tailed with the oligo C, allowing the second strand to be initiated using the oligo D primer. For example, here we have the eukaryotic oligo, mRNA with the poly A. So oligo DT primer can be used for amplifying the first strand. Then you will attach a terminal nucleotide transferase with poly C, and that C will be attached to the initial strand. So after the synthesis of the first strand, you will use a terminal transferase with C, DCTP. So that C will be attached on one end. Then you can use alkaline sucrose gradient, or you can alkaline treatment will remove that mRNA, then hydrolyze the RNA, that is alkaline treatment. Then you can use a primer that is specific for this poly C. So that is a poly G primer can be used. Using a poly G primer, the second strand is synthesized. Okay, so that is another possibility. So oligo G, reverse transcriptase, and DNTP. The second strand synthesis is using this poly G primer. So after the synthesis of the first strand, you attach a poly C tail to the first strand. Then using a poly G primer, you synthesize the second strand. And one of the advantage of this technique is that it overrules the S1 nuclease usage, which requires a trimming on one end, and that results in loss of some of the nucleotides. But if you are using this strategy, that limitation can be overruled because there is no trimming, no removal of the existing nucleotides. We are only adding poly C on one end, and using a poly G as a second primer, you can amplify the completed duplex. So here in this case, no bases or none of the part of the original mRNA is missing in the cDNA construct. So this is an improved strategy so that the complete DNA strand or the cDNA copy of the mRNA can be synthesized without any loss. Now another point is a non-directional and directional cDNA cloning. So directional cloning and non-directional cloning, that means as you know, this vector, there is a vector. For every recombinant DNA construction, we have a vector or a circularized vector that needs to be linearized by one restriction enzyme. And we have a DNA construct. The DNA construct is actually a cDNA cDNA which is copied from the corresponding mRNA. And this mRNA which actually copied is actually sometimes non-directional. Non-directional means, say, there is no difference between the two ends. So a copy can be, say, this is a DNA construct, and DNA construct can be oriented in this position or in this position. So that means just like the vector can be circularized with the DNA construct are oriented in one direction or in the other direction. So if your cloning is non-directional, that results in insertion of this DNA fragment in either direction or on both directions. Some of the clones will be inserted with the copy in one direction, and in some other copies of the clones, the fragment may be inserted on the opposite direction. So if you are placing this particular DNA fragment or cDNA copy into a vector under the control of a promoter, and the orientation of the construct is actually reversed, what happens is that starting from the promoter, it will start transcribing. And during the transcription, the entire sequence will be reversed. Maybe you know that reversal of the entire sequence or inversion of the entire sequence of the coding region will result in a completely different protein. And maybe it is not coded, or it will become non-functional, for example. So that results if the cloning is non-directional. And if your target is just to amplify the sequences and not the expression of the concerned protein and produce multiple copies of DNA or cDNA only, then for amplification purpose, the directionality doesn't matter. You can amplify the DNA in any with any orientation in any direction, so that doesn't matter. But if you are cloning this particular cDNA into an expression system so that your target is to express the concerned protein, then the directionality is more important, because it should be oriented in the correct orientation attached to the promoter. Then only the corresponding protein is coded in the correct reading frame. So that means directionality of the cloning is an important factor, and especially in expression systems. Here in this case, say we have an mRNA and the initial copy is produced, then the second strand is actually copied. And here we have a duplex DNA that is created. In this simple cloning technique, you know that there is no directionality. So it's a blended one. There is no sticky ends or any specific restriction enzymes attached on both ends. So this fragment can be randomly oriented in any direction. Okay, so such a cloning is actually non-directional cloning because there is no sticky ends attached on both ends. It's a simple cDNA construct with no specific ends. And that type of cloning is a non-directional cloning. Whereas in case of a directional cloning, it's an early strategy in which the formation of a loop at one end of the cDNA is exploited to place a specific linker. And this example, we are using SAL1 at the open end of the duplex DNA. So here, mRNA, then primer is attached, initial strand is copied. And this is the first strand loop, which is acting as a primer for the second strand copy. Then the second strand is copied. Usually, at this stage, you will use the S1 nuclease to cleave this loop. But before using that S1 nuclease to cut open this single-stranded loop region, you are attaching SAL1 linkers. So a linker is attached. Because it's a blended ligation based on the concentration of the SAL1, number of molecules of the SAL1 linkers that is provided, multiple units of the SAL1 linker, not only it will attach. It's a blended ligation. So that the mixture contains multiple units of the SAL1 or S linker. So after one after another, it will attach a few copies. So the SAL1 linkers were attached on one end, and then you are using the S1 nuclease. So the S1 nuclease will cleave the loop. So since there is a loop, the SAL1 linker is attached only on the open end and not on the other end. If you are using it in the reverse order. For example, if you are trying the S1 nuclease first and then adding solvent linkers will be added on both ends because this will be open for annealing with the S1 as a solvent linker. Okay, this loop is not cleaved initially. Linker is attached only on one end. Then subsequent cleavage with the S1 nuclease will produce a construct with one end of the construct cDNA attached with the solvent linker. Then you can again attach another linker, that is the EcoR1 linker by Bernoulli ligation. So that results in EcoR1 on both ends, added EcoR1 on both ends. Then you can cut it with the solvent and EcoR1. But in this illustration, as you know, there is something missing. Always such treatments should be done only after methylating that one. Okay, so there is a methylation treatment which is not described here. It's understood because before treating such DNA constructs with any restriction enzyme, you have to protect the restriction sites which may be present in between these sequences. So such sequences needs to be protected by methylating enzymes, and such methylases needs to be used before proceeding with such restriction enzyme digestion, and treatment of solvent and EcoR1 that will result in construction of the concerned cDNA, which is attached with the two restriction sites on two ends. So this is actually a very specific product. So that if you have the same EcoR1 on both ends, it will be oriented in any direction. But you can accordingly prepare the ends of the vector also, so that the specific direction of cloning can be maintained by two different restriction sites that is attached to the construct. And this vector can also be prepared with such specific restriction sites, and that can be specifically oriented in the desired direction during the cloning process. So this is how directional cloning is done. Clear?
Yes, sir. Clear.
Oh, okay. This is a similar strategy. But the second strand cDNA synthesis is a random branched one. That is the oligo DT primers. It carries an extension forming the solvent site. Here, you can see that there is a mRNA. Here we have a extension, oligo T primer, that is used for initial cDNA synthesis, is having an extension. The primer is actually constructed with a solvent site. Okay, the primer is having an extension. This is a chemically synthesized primer with a solvent extension. So that is attached here. So first strand is synthesized and second strand is synthesized. And on one end, we have after the complete cDNA synthesis, since the primer is actually is attached with the solvent site. So after the construction of the cDNA synthesis, we have the solvent site which is attached on one end. Then the second step, just like that of the previous example, you can use the EcoR1 linker on both ends, then cleave it with the solvent EcoR1, and the construct will have two ends with the two different restriction enzymes. That is also another method of creation of such directional cloning products with two different restriction sites attached at two ends. Now, this is a strategy from Okayama and Berg in 1982, where the mRNA is linked unidirectionally to a plasmid cloning vector prior to cDNA synthesis by virtue of a cDNA tail. You can see that this is a mRNA with a poly A tail, and this is the open vector. Here you can see the vector that is opened. Now, the annealing of these mRNA is given here. Here we have a poly T here, which is present in the vector. Now, the first strand is synthesized. You can see that the mRNA, which is attached to the poly T and poly T prime, this is a primer. This will act as a primer, and the mRNA will be copied, and using that one, the first strand is synthesized. So using the mRNA template and using this poly T, the first strand is synthesized. Then you are attaching an oligo C tail at the ends. So the oligo C will be attached on both ends of this construct and the newly synthesized strand. Then a heme 3 dijection is given. So the heme 3 dijection, which will cut at this point, and it will remove the one of the fragments with a it's a poly C and heme 3. That site is actually removed from that fragment. Then you are using an additional construct, that is a specific DNA construct with an oligonucleotide, which is having a restriction site of heme 3 and a poly G on one end. So this construct is actually chemically synthesized, and this adapter tailored with oligo G is used, and it will bind here. So this heme 3 site is here, and using that adapter with the heme 3 joined with poly G on one end, that will sit on here. So this will enable the circularization. For example, here we have the poly C at one end and poly G with the heme 3. This adapter will join or circularize this vector with the initial strand of the initial copy of the DNA. Then we have the dotted line represented here, that is the mRNA. So that needs to be removed by RNA switch, and subsequently that gap is filled with the second strand synthesis, and the second strand is finally synthesized, completing the circularization of the vector with the desired insert. So this is one of the methods that is used for a specific directional cloning, and that is a strategy developed in 1982 by Occam and Burr. So there are some drawbacks for obtaining the full-length cDNA, and that challenges were like two drawbacks of the conventional approaches is listed here. Here, where oligo DT primers used to initiate the first strand synthesis, there generally a 3' bias or preferential recovery of the clones representing the 3' end of the cDNA may be there. So previously, I mentioned that incomplete synthesis of the strands result in shorter strands. For example, out of the 750 base pair length of the mRNA, maybe the premature termination of the copying results in 500 base pair product, which will produce only the 3' end of the mRNA. Okay, so the 5' end of the mRNA may be missing because the product is actually shorter. So that's one of the shortcomings, because incomplete copy of the initial mRNA will result in shorter strands. And the second strand synthesis, if it is done, it will not represent the complete mRNA, but only a short region with a missing 5' end of the mRNA. Okay. And the size of the cDNA library, as the size of the cDNA library or cDNA increases, it becomes progressively more difficult to isolate the full-length region because of full-length clones. Because with, for example, a 500 or 600 base pair mRNA, okay, that maybe it is easy to copy in full length. But if it is a longer one, then with the increase in size of the mRNA, such premature termination and complete copying will become progressively difficult, or more and more difficult. And here we have some solutions, some strategies for that, which includes the capture method. Capture method is a method that's used for full-length cDNA cloning using eukaryotic initiation factor, EIF4E. So eukaryotic elongation factor 4E is used for performing this capture method, which results in the production of full-length mRNA, full-length cDNA. As you know, for all eukaryotic mRNAs, there is a poly A tail and a cap, 5' cap at one end. Okay, this 5' cap feature is used in this technique. So for the first strand cDNA synthesis, there are two outcomes. There may be two expected outcomes. One is a complete synthesis of the first strand, so which results in full-length synthesis of the first strand. So this is useful for us. And the second one is, as we explained, this is a premature termination, which results in a shorter first strand, and this needs to be eliminated. This should not be incorporated into the cDNA library as a representative of this concerned mRNA, because we want to eliminate this one. It is a partial length cDNA. It's not a full length one. This needs to be excluded. And for which you are using an RNase A treatment. RNase A treatment is used for cleaving the RNA, single-stranded RNA. So RNase A treatment will not have any effect if you have the first strand completely synthesized and protecting the mRNA, because it's a double-strand hybrid. So if the full-length cDNA copy is already prepared, then the mRNA is protected from RNase A treatment because there is an RNA-DNA hybrid, so there is no free RNA for lysis with RNase A. Whereas in this case, there is a free region of the RNA without any hybrid formation because of the incomplete copying that results in the RNase treatment of this region, and there will be a shorter strand produced. So you can again isolate this particular structure by and of course the 5' cap is also retained here because there is the entire molecule is protected from RNase treatment, whereas the RNase treatment will remove the 5' cap and the single-stranded region of the RNA in the second case. So you are using an affinity column, just like that of the previous poly A, poly U, saccharose or cellulose columns, which is used for isolation of the mRNA. You are using a column which is packed with the eukaryotic initiation factor 4E affinity column, beads which is attached with the 4E, and that 4E will have a specific affinity for the cap, mRNA cap, and that cap will be specifically recognized by the 4E in the column. So the column is packed with the beads are packed with the eukaryotic initiation factor 4E, which will bind with the cap, and this mRNA, the full-length mRNA, will be withheld in the separation column, and the other items will be washed away. So during the elution process, the product will be all full-length mRNAs with the complete DNA strand, initial copies of the cDNA strand synthesized. And the subsequent steps can be again performed from this eluted cDNA, so that only full-length cDNAs were prepared from this mixture, not the partial ones, because this will not be attached to the column because there is no cap. So there is no specific affinity. In the affinity column, it will be initially washed away. And the select mRNAs with the caps protected from the RNase A digestion by a complementary DNA strand called that hybrid. So this is about the capture method. Is it clear? Hello? Hello?
Yes.
Clear answer.
Yes. I want to confirm that we are all of you are online. Okay. So this is another one, which the oligo capping, and here you can see that there is a sequential treatment of the enzymes, that is alkaline phosphatase and acid pyrophosphatase. Here is the mRNA, full-length mRNA with the poly A and cap, and the partial mRNA, if any, produced, then we will have such a structure. The initial alkaline phosphatase treatment will result in the removal of a phosphate and with an OH. So 5' during the treatment of the cuttings, we discussed their treatment with the alkaline phosphatase, which results in the removal of the 5' phosphate and addition of an OH. So the alkaline phosphatase treatment results in OH addition of this point. Whereas since there is a cap, it is protected. So this phosphate is not affected by the alkaline phosphatase treatment because the cap of the full-length mRNA is protecting that from alkaline phosphatase treatment and removal of the phosphate by the OH. So whereas the partial length mRNA, if you have, is a broken mRNA, and this broken mRNA it should not be copied, as you know, because a fragment of the mRNA or a partial mRNA cannot be copied and used for the cloning, because you want to eliminate it because it's a partial one. And this can be recognized because there is a missing cap, and that cap will protect the initial one from the alkaline phosphatase treatment, whereas partial length mRNAs, if any, will be subjected to the alkaline phosphatase treatment, and a phosphate will, 5' phosphate will be removed and an OH will be added. Then the second enzyme, that is acid pyrophosphatase, is used. The second case, the acid pyrophosphatase will remove the 5' cap, so leaving the phosphate here. Then oligo, and as you know, since this sequential treatment results in a 5' phosphate at this point, which can be linked with the an oligo cap. For example, you can use a poly G or poly C, whatever, an oligonucleotide DNA fragment can be attached to this phosphate. So that phosphate addition is done here, but not here because there is an OH which is present here. So this cannot be linked with a incoming nucleotide with an OH. For example, a poly G with a free OH can pair with this phosphate and can be joined here. Whereas a poly G with a 3' OH cannot join to this OH because OH joining is not possible. So only the oligo plus addition will add a new nucleotide like a poly G or poly C or a oligo tail can be added only to the full length mRNA, not to this point, because the initial treatment of the alkaline phosphatase actually removes the phosphate. So the second stage after the cap removal, when we are adding the pyrophosphatase, this phosphatase is created and this will be joined with the oligo cap. And now the first strand is synthesized using the poly T, and the second strand can be synthesized. For example, you are adding poly G here, then poly C can be used for a specific amplification of the second strand. So second strand can be amplified using the complementary primer that is used or against the sequence that is attached here. Okay, so that is done. But in this case, it is not possible because the strand is not attached with the second primer. This cannot be amplified because there is no second primer here. The first strand can be synthesized and it can be amplified, whereas the second strand cannot be amplified because there is no complementary primer sequence attached at the end. So this is one of the method of oligo capping and sequential treatment with alkaline phosphatase and acid pyrophosphatase. This is useful where you want to confirm that your mRNA is of full length. Whereas in the previous case, this is actually a full length cDNA, and you are just copying that one. But if the mRNA itself is actually a shorter one, a partial length one, that can be excluded using this technique. Only full length, which is presented with a cap, is used for cDNA preparation and cloning. So this is about cDNA preparation and its various strategies. Any doubts?
No, sir.